Before we begin discussing the immune system, let's get a little bit of terminology out of the way up front. Immunology is the study of the immune system. Most of the time when someone is referring to immunology, they're referring to the specific ways that the body defends itself, and we'll learn more about specific defense. A pathogen is a bacteria or a virus that can cause disease to happen in the body. So disease-causing organism is a pathogen. Straightforward. An antigen is a protein or a glycoprotein that is embedded on a cell membrane. All of our body cells have antigens. Bacterial cells have antigens. Your cells have different antigens than my cells do. So the immune system recognizes foreign or self antigens and then determines whether to mount a response to destroy that cell. One of the ways that the immune system can respond is to create antibodies that bind to very specific antigens. So an antibody is created by the immune system for and in response to a very specific antigen. For instance, and we'll make this very simple here, if you have a bacteria that invades your body and it has purple square antigens, then your immune system will respond by making antibodies, proteins, that have a binding site for purple square antigens. Now, yeah, of course it's more complicated than that, but this is the same thing we saw when we looked at blood typing and the different antigens, the A, B, and Rh antigens that were on the erythrocytes and the antibodies that were in the blood plasma. Non-self is a cell or a microbe or anything that your immune system does not identify as belonging to you, and of course self is not seen as a threat because your immune system recognizes that those antigens on that cell belong to you. So the immune system is a functional system rather than an organ system. The immune system consists of cells and proteins and enzymes that have specific jobs to keep your body from falling prey to disease, from microbes, from pathogens. It can function directly where cells directly attack the pathogen or indirectly by mobilizing chemical pathways and creating antibodies. There are two defense systems that are a part of the immune system. The innate or non-specific defense that you're born with, and that includes your first and second lines of defense. They're always ready, they're always present, and they always provide the same type of defense. Your adaptive or specific immune system is your third line of defense, and it has to be primed. In other words, it has to recognize that there is a specific antigen on a specific pathogen and then mount a response according to that. So let's use an analogy to think about this. Your body is a castle, right? So let's think about a castle. What is a castle? It's a building. Around that building you have walls. You have a moat, perhaps filled with dragons or alligators, right? So what's the purpose of the moat and the wall? To defend the castle, to prevent things from getting in, very much like your skin and other structures and other chemicals try to prevent things from getting in your body. So that's our first line of defense, prevent things from getting in. The second line of defense is what happens if something does get in. All right, let's destroy it, but it's still not specific. We attack everything the same way. So think of this perhaps maybe as the alarm system. So let's say an invader got into the castle and the alarm system goes off. What happens? The police respond. Well, their response is no different regardless what triggered the alarm, whether it's a robber, a rapist, a murderer, or perhaps just a dog tripped the alarm and caused the response. So the police response is the same. This is the way inflammation and fever and other things work in your body. So something got in, we want to stop it, but it works the same way against all invaders. But then we have that adaptive or specific line of defense, where your immune system has to recognize a specific pathogen and mount a specific response that will be effective against that pathogen. So let's say we go back to our castle analogy. So the police respond just like always. The invader got in, breached the wall and the moat, so he got through the first line of defense. He triggered the alarm system and the police responded, so he got into the second line of defense. Police responded to try to take him out. But what if that doesn't work and that pathogen, that invader, gets in and starts causing the immune system to have to mount a response to prevent disease or to get rid of the pathogen before further disease? So same thing with our castle. The police responded, but now the invader has taken a hostage. So what's going to happen if there's a hostage situation? You call in a certain response team, but you call in a negotiator which has a specific job to try to fix the situation. What if the invader has a bomb? Then you've got to call in the SWAT team and the bomb team, right? So your response was different based on what happened in response to that invader. So that's the way your immune system works as well. It mounts a specific response based on a specific threat. Innate or non-specific immunity includes the external, physical, and chemical barriers that our skin and mucous membranes provide for us. But it also includes some internal defenses, such as antimicrobial substances and natural killer cells, phagocytes, inflammation, and fever. So we'll begin by looking at the first line of defense, which are the surface barriers. The skin and the mucous membranes of the body are part of the first line of defense against those pathogens getting in. So the skin and the mucous membranes provide both a physical and chemical barriers that discourage pathogens and foreign substances from being able to penetrate and get into the body and cause disease. Think about when you learned about the skin and you saw there were so many layers of those very tightly packed together keratinized cells forming the epithelial layer of the skin, right? The epidermis. So that's a very tough physical barrier that prevents most microbes from getting in. But what else happens? We're constantly shedding those epidermal layers. So the microbes that are on the surface of the skin get shed away with the dead skin cells. So bacteria very rarely penetrate the intact surface of a healthy epidermis. But as we know, that surface doesn't always stay intact, right? We have cuts and burns and punctures and different types of wounds that allow those pathogens to penetrate the epidermis, and then they can get into the adjacent tissues or even circulate in the blood to other parts of the body. The epithelial layer of mucous membranes, remember mucous membranes line open body cavities. Your mucous membranes secrete that sticky, thick mucus that lubricates and moisturizes cavities. But because it is thick and gooey, it traps a lot of microbes and foreign substances. The mucous membrane of your nose has mucus-coated hairs. Yes, you have nose hair for a reason. The hairs and the mucus trap and filter out microbes and dust and pollen and pollutants from all the things that we breathe in the air around us. The mucous membranes of the respiratory system, the upper part of the respiratory system, contain cilia. Remember, cilia are those little microscopic hair-like projections that are found on some of our epithelial tissues, and they beat in the liquids of the mucus all the time, and as they do, they're propelling dust and microbes that are trapped in that mucus back up toward the throat. So we swallow that mucus then as a part of our saliva. The pathogens then go down to the stomach and the gastric juice destroys them. Sometimes we also cough or sneeze if we have a buildup of mucus, and then all the trapped pathogens that are in that mucus get moved up to the mouth as well. We either spit it or we swallow it, right? But there are other fluids that you might not think of that protect your body. Your lacrimal apparatus, which has your lacrimal glands in your eyes, makes tears, and those tears, when we blink, spreads across the surface of the eyeball, so it's just continually washing away microbes that may be on the surface of the eye and keeping them from settling. The The tears also contain an enzyme called lysozyme. To lyse means to break. Zyme is referring to enzyme. So lysozyme helps to break down cell walls in certain bacteria. You also have lysozyme present in your saliva, your perspiration or your sweat, secretions in the nose, and some of your tissue fluids. Saliva also moisturizes and washes the mouth, very similar to what we see with the flow of the tears on the eyes. Urine through the urethra washes out microbes that may be in the urinary system. Vaginal secretions keep the vagina moist, and as it flows out, it also moves things out of the vaginal canal. But vaginal secretions are also very high or very acidic, so very low in pH. So that helps to inhibit bacteria other than your normal bacteria from being able to colonize in the vagina. In response to some microbes, your body just gets rid of it, right? Have you ever thought about what happens when you eat something that may contain spoiled food, such as the botulism toxin from bacteria? What happens? It triggers your gastrointestinal tract to react vigorously, and you throw up that food containing that toxin. Or you have a rapid onset of diarrhea so that you get rid of it from the other end, right? So your body is using the things built in to get rid of these toxins, get rid of these pathogens before they can get deeper into the body. Your oil glands in your skin secrete sebum that forms a protective filmy layer over the skin. And the acidity of the skin is very low. It's acidic, pH of about 3 to 5. So that's caused in part by the fatty acids and the lactic acids that we create that are on the surface of those cells as we are getting rid of them. Perspiration helps flush microbes off the surface of the skin. Gastric juice, as I mentioned earlier, is very, very low in pH, made up of a lot of hydrochloric acid and mucus and enzymes. So the acidity of the gastric juice destroys bacteria and most toxins. So we've got a pretty good first line of defense, right? Keep things out. But it doesn't always work. So now we've got to think about something's gotten in, what do we do? So pathogens that have penetrated the physical and chemical barriers of the skin and the mucous membranes encounter our second line of defense, which includes some antimicrobial substances, phagocytes, natural killer cells, inflammation, and fever. Phagocytes are specialized cells that perform phagocytosis, or eating, the ingestion of microbes or other particles such as cellular debris. We have two major types of phagocytes, and those are neutrophils and macrophages. So when an infection occurs, neutrophils and monocytes migrate to the infected area. During this time of migration, the monocytes enlarge and they get out of the bloodstream into the tissues and become phagocytic macrophages. We call those wandering macrophages. Eosinophils are weak phagocytes, but they are primarily responsible for helping to get rid of parasitic worms. The neutrophils become phagocytic once they encounter that foreign substance. Those neutrophils are your rapid responders. They're the first one on the scene. When phagocytes are being mobilized, we have an increased number of the immune system cells that are going to come into that area. So you have a lot of these immune cells rushing into that area of damage, and they do so because of chemicals that are released that cause chemotaxis. So the increased number of cells and increased chemicals such as leukocytosis-inducing factors are going to cause a lot of cells, right? So leukocytosis happens quickly. Chemotaxis, those damaged cells release inflammatory chemicals that attract more and more cells into that area, resulting in the leukocytosis. Neutrophils travel to the inflamed area and they stick to the capillary walls in an action called margination. And then those neutrophils have to squeeze out of the blood vessels if they're going to get out to the damaged tissue. So that action is called diapedesis. So the neutrophils start inserting little pieces of their cell bodies out through the spaces in the capillary walls, and through diapedesis they move out into those tissues. Phagocytosis is broken into 5 different phases. Chemotaxis begins when those damaged tissues release chemicals. That chemical stimulates the movement of the phagocytes to the area of the damage. So they attract the phagocytes by releasing those chemicals. Those chemicals could come from the damaged cells, from the invading microbes, or the activation of complement proteins, which we'll see in a minute is a part of our inflammatory response. Once chemotaxis occurs, we've secreted the chemicals to attract the cells, then adherence is the next step. So the attachment of the phagocyte to that invading microbe, or whatever the foreign material is called adherence. The binding of complement proteins to the invading pathogen enhances adherence. Again, we'll talk more about complement, but it's just a group of enzymatic actions that are driven by a group of proteins that increase the inflammatory and immune response. The third step is ingestion. So the cell membrane of the phagocyte extends out little cytoplasmic extensions called pseudopods that engulf the microbe in a process called, simply enough, ingestion. When the pseudopods meet, so they've completely surrounded the microorganism with a sac, that little sac is called a phagosome. Okay? So remember, some means body. The fourth step, the phagosome enters the cytoplasm, bringing that pathogen in with it, and it merges inside the cell with lysosomes. Lysosomes are little bags, little vesicles that contain lytic enzymes. So the lysosomes contribute lysozyme, which breaks down the microbial cell walls. And also the lysozymes usually also have digestive enzymes that help to degrade or break down carbs and lipids and proteins and nucleic acids, so helping just to break down all the components of that microbe that is being destroyed. And then the last stage, of course, is the killing. The chemical onslaught that's provided by the lysosome and all those digestive enzymes and oxidants that are in that little bag that we create, right? So we took the phagosome and we fused it together with the lysosome. So all the stuff in there causes the quick killing of those microbes. It's very effective against many different types of microbes. So those are the steps in bringing in that microbe, that pathogen, and phagocytizing it, breaking it down, killing that pathogen. The second type of nonspecific defense that we'll look at in our second line of defense are the natural killer cells. So natural killer cells make up about 5 to 10% of the lymphocytes that are circulating in your blood and in your organs. They're also present in the spleen, the lymph nodes, the bone marrow. They have the ability to kill a wide range of infected body cells and pathogens, and even certain tumor cells. Natural killer cells attack any body cell that has abnormal or unusual plasma proteins, unusual antigens. So the binding of a natural killer cell to a target cell, such as an infected human cell or a bacteria cell, causes the release of granules that contain toxic substances that are made in those natural killer cells. One of the most common toxic substance that the natural killer cells release is something called perforin. So perforin perforates. What do you think of with perforated paper? It has holes in it, so you can tear it out, right? So to perforate means to poke holes. So perforins insert into the plasma membrane of the target cell or the pathogen and create channels or perforations in the membrane. Because of that, extracellular fluid can flow into the target cell and intracellular fluid can flow out. So this is going to result in that cell lysing or bursting. The natural killer cells also can release granzymes, which are protein digesting enzymes that can then induce that target cell to undergo apoptosis or self-destruction. The third nonspecific second line of defense we'll examine will be the inflammatory response. I'll warn you now, this one will take a few minutes. inflammation is a nonspecific defense response to the body's tissue damage. So the conditions that can result or produce inflammation are pathogens, irritants, disturbance or distortion of cells, extreme temperature, abrasions, insect bites. Man, there's a lot of stuff we know that can cause inflammation. So inflammation is simply an attempt to dispose of the microbes or the toxins or the foreign particles at the site of the injury, to prevent them from being able to spread to other tissues, but also to prepare the body for tissue repair so that we can restore homeostasis. So injured cells are going to release chemicals. So what happens? There are certain signs or symptoms that are associated with inflammation, and you can remember these by using the acronym PRISH, P-R-I-S-H. P is for pain due to the loss or to the release of certain chemicals. R is for redness because more blood is going to rush into the affected area. I is for immobility because that sometimes results from a loss of function when you have a very severe inflammation. S is for swelling. Swelling results from an accumulation of body fluids. And H is for heat, which is also a result of more blood rushing into that infected area. Because the inflammation is one of the body's nonspecific defense mechanisms, the response is the same, whether it's to a small cut or major tissue damage as the result of a burn or radiation, or it's the same with a bacterial or viral infection. In every case, the inflammatory response has the same three basic steps: vasodilation and increased permeability of the blood vessels, immigration or movement of phagocytes from the blood into the interstitial fluid, and then tissue repair. Two things happen immediately in response to a pathogen when you're going to have an inflammatory response, and these occur within blood vessels in that region of tissue injury. Vasodilation, or an increase in the diameter of the blood vessels in that area, and increased permeability of the capillaries. So increased permeability means that things that are normally kept within the blood are able to pass out of the blood from those capillaries into the tissue. Vasodilation allows more blood to flow through the damaged area, and the increased permeability permits proteins such as antibodies and clotting factors to get out into the injured area from the blood. So the increased blood flow can also help to remove some of those toxins and dead cells from the area of the damages. There's some different substances that are released, different chemicals that contribute to vasodilation and increased permeability, but also to other aspects of the inflammatory response. Histamine is one of those chemicals. It's released in response to the injury. So mast cells in connective tissues, basophils, and platelets in the blood release histamine. Neutrophils and macrophages are then going to be attracted to the site, and they also stimulate the release of more histamine. So what does histamine do? It causes vasodilation and increased permeability of blood vessels. Kinins are chemotactic agents. So kinins cause chemotaxis to occur. So kinins are chemicals that are released from, or they're actually formed as inactive proteins in the blood, but they're released during tissue damage, so they attract phagocytes into that area. Prostaglandins intensify the action of histamine and kinins. They also help to stimulate immigration of phagocytes through the capillary walls, so the phagocytes get out to the damaged tissue. So prostaglandins are synergistic. They increase or enhance the effect of other chemicals. Leukotrienes are released by basophils and mast cells. They increase permeability. You get the picture here, right? A lot of things going on to increase permeability and attract cells. They also help the phagocytes in adherence to the walls so that they can get out to those pathogens and to the damaged areas. Leukotrienes also increase chemotaxis. And then there's complement. Complement is a group of proteins that form a system that enhances the inflammatory response. We'll talk a little bit more about that as well. So the dilation of the arterioles and the increased permeability of the capillaries produce 3 of the signs and symptoms of inflammation: heat, redness, and swelling. Heat and redness result from the large amount of blood that accumulates in the damaged area. With the blood, you have an increase in temperature. Okay, so temperature increases in that area, redness, swelling. As the local temperature rises, metabolic reactions are stimulated to occur more rapidly. So the increase in temperature increases the rate that enzymes work, and even that causes additional heat to be released because the metabolism is higher in that area. Edema results because of the increased permeability of the blood vessels, so more fluid is leaking out from the blood plasma into the tissue spaces. Pain is one of the main symptoms of inflammation. It results from injury to the neurons in that area, but also from the toxic chemicals that are being released from the microbes. Kinins can affect the nerve endings and cause a lot of pain as a part of inflammation. Prostaglandins intensify and prolong the pain. So not only do they help to increase the response, but they also increase the pain. The pain can also be due just to that increase in pressure from the edema in that area. Although neutrophils, remember, the rapid responders, predominate the early stages of an infection or inflammation, they die off pretty rapidly. As the inflammatory response continues, monocytes follow the neutrophils into the infected area. Once in the tissue, monocytes become wandering macrophages. The macrophages are much more potent phagocytes, as you would expect, than the neutrophils. So they're large enough to engulf damaged tissue, worn out neutrophils, and the invading microbes. But eventually the macrophages are going to die also. So within a few days, a pocket of dead macrophages, damaged tissue, dead phagocytes, neutrophils, all of this forms a pocket of what we call pus. So dead cells and fluid. Pus formation occurs in most inflammatory responses and usually continues until the infection is gone. Sometimes, though, the pus reaches the surface of the body or drains into an internal cavity, and when it does so, the body just disperses it and gets rid of it. Sometimes, though, the pus remains even after the infection has been healed. The infection is gone. In these cases, the pus is very gradually destroyed over a period of several days and then resorbed. So, looking at a little more information on some terminology here, inflammation can be classified as acute or chronic depending on several factors. In acute inflammation, the signs and symptoms develop rapidly and usually last for a few days or even a few weeks. It's usually mild, self-limiting, and the principal defensive cells are the neutrophils. Examples of an acute inflammation are sore throat, appendicitis, a cold or the flu, bacterial pneumonia, or even just a scratch on the skin. In chronic inflammation, the signs and the symptoms develop more slowly and can last up to several months or even years. It's often severe and progressive, and the principal defense cells are the monocytes and the macrophages. Some examples of chronic inflammation include mononucleosis that we talked about recently, peptic ulcers, tuberculosis, rheumatoid arthritis, and ulcerative colitis. So let's think back about that pus again. So pus, remember, is accumulations of fluid and dead, damaged cells. Okay? So if that pus can't drain out of an inflamed area, the result is an abscess. So an abscess is an accumulation of pus in a confined space. So here you can see pus very obviously draining out. Okay? This is an abscess, very commonly seen in the mouth, or one of the more common places to see an abscess, where the pus accumulates and it does not drain out. A lot of times you do have to visit a dentist to have these drained, and then, of course, some antibiotics can help with the healing process. Some examples of accumulations of pus: pimples, boils. What do we usually do? We try to squeeze on them and get that pus out, right? Be careful when you're doing that. You're opening a body area and your fingers are covered with bacteria. nice little breach in that first line of defense, right? When the superficial part of that inflamed tissue sloughs off on the surface of an organ or a tissue, the result can be an open sore that we call an ulcer. So people with poor circulation, for instance, those that have diabetes mellitus or advanced atherosclerosis, are susceptible to ulcers in the tissues of their legs. A lot of times people with diabetic conditions will develop these ulcers on their feet as well. So these develop because of poor oxygen and nutrient supply to that tissue, and then it becomes very susceptible to even to a very, very mild injury or an infection. When these go untreated and uncared for, it's not uncommon for a person with a diabetic condition and ulcers of the feet to lose toes or even lose the entire foot. Continuing to look at the nonspecific defenses, and remember we're on the second line of defense, the fourth will be antimicrobial substances that discourage or prevent microbial growth. So lymphocytes, macrophages, and fibroblasts that are infected with viruses can produce things called interferons. So these are proteins that once they're released by the viral infected cells, they diffuse into unaffected neighboring cells, and they induce the synthesis of antiviral proteins, including enzyme PKR, which is a protein kinase. It's an enzyme, a protein that replicates with DNA replication. So interferons interfere with DNA replication. Viral cells cannot create new viral cells unless they can invade cells, our cells, and replicate their DNA. So viruses can only cause disease if they can replicate within body cells. Interferons prevent that from happening. So we do now have the ability in research labs and in medical labs to genetically engineer interferons that can be used to successfully treat certain types of viral infections such as hepatitis C, herpes, and some viral infections that may be a part of organ transplants. The second antimicrobial protein we want to look at is that group of complement. Remember, I mentioned this earlier. So complement is 20 to 30 different proteins in the body. So normally they're inactive proteins in the blood plasma and on the surface of cell membranes. So they make up the complement system. But when they're activated as a part of the inflammatory response, these proteins complement or enhance immune system reactions. So the complement system can cause cytolysis or bursting of microbes, promote phagocytosis, help antibodies to be created and to cause destruction of cells. And all of this, of course, is going to contribute to the inflammatory response. Another really cool thing about the complement system is these proteins act in a cascade, so very much like a domino effect, where one reaction triggers another reaction, which triggers another reaction, and so on and so forth. And each succeeding reaction causes more and more and more product to be formed in that inflammatory response. So the net effect is amplified many, many times more than it was when the complement first began. So complement can cause membrane attack complexes that allows the immune system to recognize that these cells need to be destroyed and proteins such as perforins to be created. It also can cause opsonization, which causes the membranes to become sticky so that adherence can happen much more easily. Remember, adherence is where those phagocytes adhere to the walls of the cells and adhere to the damaged tissues and get out there so they can clean that damage up. And the final non-specific second line of defense that we want to talk about is fever. We've all had fevers, right? What happens? You feel achy, you feel tired, your temperature goes up. Why did it go up? Because there's a pathogen in your body and your immune system is responding to that by creating an abnormally high body temperature. So the hypothalamus has a thermostat, and that thermostat gets knocked out of whack, so it's reset, and body temperature raises. So this commonly occurs during infection and inflammation. There are many bacterial toxins that can cause an elevation in body temperature, sometimes by causing the release of fever-causing cytokines or pyrogens. Elevated body temperature has the effect of intensifying the effect of interferons, interfere with viral replication, also inhibiting the growth of some microbes, and increasing metabolism, so it speeds up reactions that can aid in the repair process. So if you have a low-grade fever, you're an adult in normal, healthy condition. A low-grade fever is simply your body's immune system trying to get rid of whatever is causing that fever. So maybe you shouldn't rush out and take something to decrease your fever, as long as you are healthy otherwise, right? And you've not been exposed to something that you know could cause you to be really, really sick, right? But in young children, in babies, in the elderly, in those that have other conditions that may affect their immune system, a low-grade fever can be very harmful. So you want to be careful. Consider the overall condition of the person with the fever. And, of course, consult your doctor.